Hello, everyone. Welcome to our first podcast entirely dedicated to the project Cure4Aqua, which is about developing new approaches to prevent aquatic fish diseases and prioritizing fish welfare in aquaculture production. Cure4Aqua launched in November 2022. It will last 4.5 years with a budget of 4.8 million euros that is funded by the European Union. And it involves a multinational and interdisciplinary team of 31 partners from 16 different countries, integrating researchers, academics, and industry stakeholders. Today, we will be interviewing Ivona Mladineo, head of the Laboratory of Functional Helmintology in the Biology Center of the SEC Academy of Sciences, and also the project coordinator of Cure4Aqua. Ivona is going to share some insights into the project's groundbreaking approaches and goals. So just welcome, Ivona.
Thank you, Carla.
So just could you start just by giving us an overview of the Cure4Aqua project and its main objectives?
So Cure4Aqua is a research and innovation action coordinated by Biology Center in Czech Republic and involves 30 partners from 15 different countries, including 10 EU member states and also UK, Switzerland, Norway, Thailand, and Chile. So we are even more broader than just EU. All the Cure4Aqua will support the entire EU aquaculture industry. We will focus on species such as the rainbow trout, gilted sea bream, European sea bass, greater amberjack, Senegalese sole, meager, and common carp. And originally, the idea was that the project will be driven by the understanding that the European aquaculture industry needs to evolve in order to meeting the growing demand for high-quality sustainable seafood. So, Cure4Aqua seeks to address several challenges that are facing the European aquaculture industry, including slow growth, disease control, climate change, antimicrobial resistance, and ethical and environmental standards. So, our goal is to engage stakeholders from all target groups and improve the resilience of EU aquaculture by developing cost-effective vaccines to prevent fish diseases, identifying markers with diagnostic capacity, developing alternatives to antibiotics, and develop new tools and technologies to improve fish welfare monitoring.
Wow, that sounds amazing. And so what are some specific diseases or pathogens that Cure4Aqua was focusing on? And can you just tell us a bit more about their implications?
Yes, so we are focusing on a variety of diseases and pathogens that affect both marine and freshwater fish aquaculture. So we are targeting specific pathogens for vaccine development, including two bacterial, two viral diseases, and the parasites. So for the viruses, we are targeting nervous necrosis virus and carpedema virus, and for parasites, primarily Tetracapsuloides bryosalmone. Cure4Aqua will also include marine pathogens such as Vibrio species, P.C. rickettsia, Midichlorians, and Chlamydia, and also the parasites Paricotyle chrysophryi, while freshwater pathogens include viruses as koi herpes virus, infectious pancreatic necrosis virus, and bacterial Flavobacterium, and again the parasite Tetracapsuloides and Sphaerospora molnari. However, the selection of these specific diseases and pathogens was based on the importance of implications that they have for the industry and consumers. And unfortunately, we do not have an accurate idea about the financial losses for specific diseases of pathogens. Therefore, we are working on developing what is called burden of disease or BOD analysis. And this is a process that involves summing up the direct and indirect financial losses accounted to a disease incidence in the farm and calculating how much that specific disease burdens the farmer. Interestingly, these analyses are commonly done in human medicine, but has been difficult to introduce this concept in aquaculture due to many parameters that need to be incorporated in the calculus and that are usually not publicly available.
That's very interesting, the concept of burden of disease and how it's going to be used in the project. So how exactly does Cure4Aqua fit into this broader picture of sustainable aquaculture, and what are actually the challenges that the project is addressing?
So we fit in this broader picture of sustainable aquaculture by directly addressing key challenges hindering the industry's growth and environmental responsibility. So the European aquaculture industry currently faces several obstacles. For first, the slow growth compared to global trends, and we know that EU aquaculture industry has not kept pace with the global aquaculture growth, lagging behind since 2007. Then an effective and cost-efficient control of pathogens is crucial. We are tackling this through vaccine development for 5 key pathogens that I mentioned previously, also alongside alternative bio-based solutions like probiotics and antimicrobial peptides. Then, of course, there is the climate change and antimicrobial resistance that continues to be a challenge. And these unpredictable threats demand innovative solutions. So we are investigating environmental disease interactions and seeking alternatives to antibiotics and therefore mitigating also the antimicrobial resistant risks. And last but not least, also the ethical and environmental standards that we want to address because consumers today increasingly demand sustainable and ethically produced seafood, and Cure4Aqua emphasizes fish welfare, develops high welfare standards, and also explore the environmental impact of farming practices.
Wow, those are big challenges, of course. So now can you just discuss the innovative approaches that the project is actually tackling to use these current health and welfare challenges?
Cure4Aqua uses several innovative approaches to achieve its quite ambitious goals. Some key innovative approaches employed by Cure4Aqua are, for example, vaccine development that aims to address the lack of appropriate commercial vaccines for the EU aquaculture industry. And here the goal is to deliver vaccines for 5 emerging fish pathogens. This vaccine will incorporate diverse formulations, including inactivated wall pathogen vaccines, which are kind of standard, recombinant protein vaccines, mRNA vaccines, and the dodecine vaccine. And additionally, we are also exploring alternatives to traditional injection methods in vaccination, such as use of a deep oral administration and live feed system for mucosal delivery. So these approaches aim to improve vaccine efficiency and also reduce the stress on fish. And then we are also focusing on epigenetic programming and microbiome modulation to improve fish resilience to diseases and further enhance their welfare. This epigenetic programming involves developing rearing and management protocols that would reduce the stress, improve welfare, and boost immunity through epigenetic and microbiome modifications. The project also uses advanced technologies, of course, like omics, to identify the best markers representative of immunocompetence and welfare. And we hope that this will eventually lead to the creation of epimarker panels for use in selective breeding programs to enhance disease resistance and stress tolerance. And yes, again, Cure4Aqua also addresses the urgent need for antibiotic alternatives by exploring effective bio-based solutions such as probiotics, phage therapy, antimicrobial peptides, and recombinant antibodies to combat fish diseases. To further enhance biosecurity, we will also develop comprehensive codes of good practice and a scoring system for assessing biosecurity measures at both farm and company levels. And as mentioned previously, Cure4Aqua also uses a multi-actor approach, which includes forming process groups, what we call PGs, with representatives from research, industry, policy, and consumer organization. And these groups are involved in various stages of the project in order to help us to decide the best directions.
Nice. And could you also dive a little bit into the role of early disease detection in aquaculture and how Cure4Aqua is advancing in this area?
So fish health is crucial for aquaculture industry, and early detection of diseases is essential for preventing widespread outbreaks, to minimize also the economic losses, and of course to ensure fish welfare. So the faster a disease is detected, the quicker appropriate measures can be implemented to mitigate its impact. And we are working on early disease detection through development of new advanced monitoring and diagnostic tools. For first, there is Cure4Aqua developing non-invasive monitoring tools to assess fish health and welfare. This involves using underwater cameras and deep learning algorithms to monitor swimming behavior and growth, then stereo cameras to monitor exterior skin and fin, eye, and operculum conditions, and finally, also the sensors to collect environmental data like temperature, dissolved oxygen, salinity, and water current, so to integrate all these biological and abiotic parameter in one quite precise tool. Then the project also uses advanced technologies to develop rapid and efficient diagnostic tools, aptamers, which is quite an innovation in aquaculture, as an alternative to antibody-based methods for developing new markers for pathogens and fish immune parameters. Then also 3D bioprinting to create culture and infection system, reducing the reliance on live animals for testing within the experiments. And Cure4Aqua is also working on the development of qPCR on chip technology for rapid and sensitive pathogen detection. And this technology allows for non-invasive sampling from water, skin, and sera, reducing the need for lethal sampling methods.
Wow, that all sounds very interesting and very innovative. So you actually mentioned earlier the development of alternative treatments. So what exact treatments are being explored in the project to replace these traditional?
This is very timely now, and Cure4Aqua will develop new combined probiotics and phage applications to control first Vibrio and Flavobacterium in European sea bass and rainbow trout. And this approach aims to promote a healthy microbiome and protect fish during early rearing stages. The project will investigate the genome sequencing and phenotypic analysis of probiotics phage resistant bacteria to understand the mechanisms and fitness cost of resistance. Then Cure4Aqua will also develop a novel antimicrobial feed additive using nano-encapsulated antimicrobial peptides. The additive will protect European sea bass from gram-negative bacteria during the rearing cycle. And the project builds on previous research by Biology Center that characterized parasite-originating antimicrobial peptides with potent bactericidal effects and no host cyto or genotoxicity. And finally, for alternative treatments, we will also explore passive immunization, and we will use virus neutralizing recombinant antibodies. So the project will specifically develop optimized protocol for passive immunization of the European sea bass and Senegalese sole against nervous necrosis virus.
Okay. And you also mentioned that fish welfare is a priority for the project, right? So could you elaborate a little bit on how the project integrates fish welfare into its research and solutions?
So, fish welfare is standing quite important in our agenda. So, the project recognizes the growing importance of fish welfare in aquaculture and aims to improve it through a multidisciplinary approach. So, this includes developing first a welfare score index for key species, creating what we call a quality of life scaling system for informed interventions, and developing biosecurity protocols to minimize disease outbreaks. Then we also focus on non-invasive monitoring tools like machine vision that I mentioned previously, using artificial intelligence to assess fish welfare without causing stress. Recognizing the interconnectedness of human and animal well-being, we will also investigate the relationship between farmer well-being, fish welfare, and company growth, which I think, to my knowledge, has not been done before. And so by integrating fish welfare consideration into Cure4Aqua research, we will contribute to a more ethical and sustainable aquaculture industry. This will not only enhance the well-being of farm fish, but also increase consumer confidence in European aquaculture products and promote a more responsible and more sustainable approach to food production.
Okay. So far, what have been some of the significant milestones or findings that the project has received?
The significant milestones have been practically reached in each of Cure4Aqua work packages. What I consider very important is that we have created process groups that consist of a number of stakeholders that are involved outside the consortium to help us steering the research towards a more useful innovation. I will tackle randomly a few research-based milestones that we have now by the end of the first half of the project and that are on the pathway of becoming important, maybe marketable outcomes. So, for example, many bacterial isolates have been sequenced, and this will help us in reverse vaccinology, a process where we look first for the ideal vaccine candidates in pathogens genetic material, and preliminary results of using phages and probiotic bacteria in early fish developmental stage to encourage a lifelong fish resilience are supporting our hypothesis that fish can be epigenetically programmed and that maybe some of these changes can be traced genetically as markers in the upcoming generations. Then new algorithms have been developed and implemented in remote monitoring system installed in sea bream and sea bass cages that help us track the feeding of fish, whether they are growing well, how they are swimming or breathing, whether they have damage on the skin or fins. With installed environmental sensors in those cages, it is amazing the amount of data that we are receiving and from which we can draw out extremely useful information of fish well-being. But if a farmer is, for example, indecisive whether to implement a new monitoring system or, for example, a new vaccine due to foreseen expenses, he can rely on Cure4Aqua economic model. This model, which is going to be gamified for friendly use, takes into account baseline cost and profits of the farm and stimulates farm's business in a scenario where the monitoring system has been bought and installed or a vaccine applied, which means that it gives an estimation of the time needed for the farm to start make more profit after implementing the intervention, which in this case would be vaccination or monitoring. And again, I would like to mention the antimicrobial peptide that comes from a parasitic nematode, but it is able to find all major aquaculture gram-negative bacteria. To achieve targeted dosage, we have put it in a natural polymeric nano capsule so that it is absorbed within fish body and released at the site of pathogen infection. And this has been done in collaboration with the Thai partner. For the rest that I have not mentioned, please visit our website or track us at the upcoming conference of the European Association of Fish Pathologists that it's going to be organized in beautiful Heraklion in Crete on 1st of September this year.
Wow, that seems all truly revolutionary, and it seems like a very exciting time for Cure4Aqua. So just finally, as we look towards the future, what are the next steps for Cure4Aqua, and how do you envision the role of projects like Cure4Aqua in shaping the whole future of aquaculture?
So within 4.5 years of Cure4Aqua life cycle, we are busy focusing on delivering the activities, but I believe that the real impact of the project will emerge after this period, once we deliver our innovative solution, publish the results, and observe the in-field implementation of the outputs. We will probably grow more exciting collaborations outside the consortium, interact and join new consortia, bringing our newly gained experiences. And we already have such cases like European Reference Center for Aquatic Animal Welfare called AQUA, or the EU Partnership on Animal Health and Welfare, where a couple of our team partners are actually taking important leadership parts in these consortia. So I think of that fondly as Cure4Aqua ramification and maybe reincarnation, but whether we set some new baseline ground for further research or launch a new diagnostic tool on the market, I hope we will achieve a positive shift in thinking about aquaculture in the mind not only of a farmer, but also a consumer, a welfare activist, and policymaker, because at the end of the day, we all live off and live for sustainable aquaculture.
That's totally true. Well, thank you very much, Ivona, for these insightful answers. We also just wanted to thank all the people that are listening to us today. As you heard today, Cure4Aqua's goal is to contribute to the revolution of European aquaculture. So stay tuned to learn more about sustainable aquaculture, health, and welfare. And just visit our website for more news. Thank you so much.
Thank you. Bye bye.